Thanks very much, Judy. I'd like to thank Rick, Hilary and Judy and the steering group for inviting me to speak to you today. What I'm going to talk to you about is Toxoplasma. The title of the talk is The World's Most Successful Parasite. You may or may not agree with me at the end of the talk what you think of this. But what I want to cover in the talk is to look at some of the new research we're doing in our group at Moredun, and also incorporating different aspects of the research covering the veterinary side, the public health side, and also the environment. And I think the whole field of Toxoplasma has opened up quite a lot in the last few years. And this is largely due to a really interesting conference that was held in Brazil about 5 years ago. And it was celebrating 100 years since the discovery of Toxoplasma. And the reason this conference, I think, was so influential is there's been a lot of really interesting stuff coming out of Brazil that had been largely ignored by the European scientists and the North American scientists. And at this conference, a bit like this conference here, there was no concurrent sessions. We were all mixed up together, so you had vets, biologists, medics, public health people, all in together, and we all had to listen to each other, which was quite good. And therefore, it encouraged quite a lot of multidisciplinary thinking and learning. And we all learned a lot from each other about the parasite, which actually is one of the world's most successful parasite. It can infect all warm-blooded animals. And its impact is far-reaching across the different disciplines. So coming out of that conference, there's been a resurgence of interest in toxo. And I'm just going to talk you through some of the new stuff that we've been doing on that. So, give you a bit of context. Toxo does infect all warm-blooded animals, although the cat and members of the cat family are the only definitive hosts of the parasite. So if you look across a wide spectrum of different animals, you get very different clinical signs depending on the host species. And there's a lot of different reasons for this, some to do with the host species themselves, some to do with the parasite genotype, which I'll talk about later on, and some of it's to do with evolution, and if these species have sort of grown up alongside the cat, because the cat is critical in the life cycle here. So just to give you some examples here, if you look at this side here, on one side you've got marsupials who have evolved away from the cat, and they are very vulnerable to infection with Toxoplasma. And often you see cases in zoos where if they're infected with the parasite, they will often die of acute infection. You see this with penguins as well. So where animals have evolved away from the cat, they haven't experienced toxo, you can get very acute infections. A species we work on a lot at Moredun is sheep. It's a big problem in the veterinary world, where it causes abortion in sheep. Another intermediate hosts here are cattle, and I've got cattle in here because we always thought before cattle were not a problem with toxo and that you don't get a lot of disease in cattle. However, cattle are intermediate hosts of the parasite, and a way of transmitting the parasite to people is through eating undercooked meat that has tissue cysts of the parasite. So we know cattle can get infected, they don't get disease, so we've largely ignored them in the veterinary side. However, from the public health point of view, they're very, very important, and I'll talk about that later on. Now we've got a picture down here of cats and mice, and this is the normal situation you get with a cat and a mouse. Where the mouse is trying to get away from the cat. There's some quite interesting research going on a few years ago down in Oxford where they looked at Toxoplasma infected mice that were no longer afraid of cats. So the obvious sort of simplistic idea here is that the mouse is trying to get into the mouse, the parasite, not the mouse. Mouse is a victim here. The parasite is trying to get into the cat to complete its life cycle. And on the back of that research, a lot of people wondered what it was doing to us if we were infected with the parasite. So I'm going to take you on to the people side of things, where if we're infected with Toxoplasma, does it change your behaviour in any way? Because the parasite does sit in your brain, and what does it actually do? So there's been a resurgence of interest in Toxoplasma, having a look at the public health side of it. So if we look at the impact of the parasite, it's on the veterinary side, which we've been working on at Moredun quite a lot, it's a major cause of abortion in sheep and goats worldwide. It's the second most important diagnosed cause of abortion in sheep in the UK, across Europe, and it's really across the world. So places like New Zealand, Australia, other countries that are big sheep rearing countries, it's a major, major problem. Another high-risk group is congenital infection in women. Now, the disease in sheep and the disease in women is very similar. So there's quite a lot of interesting comparative work you can do between the two to try and learn from each other. Congenital infection in women, it's only a problem if you get a primary infection during pregnancy, and the stage of gestation is critical when you get the infection as to what the outcome will be. And it's very similar to some work we've done in sheep, very similar there. What happens is you can get very severe disease of the fetus if the woman is infected for the first time during pregnancy. And the baby can develop with eye disease, brain damage, and can have lifelong learning disability. The incidence of toxoplasma in women, pregnant women, is not that high, luckily. In UK, it's about 5 per 100,000 births. But if you look at countries like Brazil, it's much higher. It's about 5 per 1,000 births. And the impact of having a congenitally infected child is huge. And this is something I'm going to talk about later on when we look at the impact of foodborne toxo. So the other high-risk group you've got are immunocompromised people. And this is often seen in people who have a persistent infection, because once you get infected with the parasite, you stay infected for life. And what happens if your T cells or T cell immunity gets depressed, and this has happened a lot in the 1980s with HIV patients, before we now have much better antiretroviral therapies, but before that, there was a lot of deaths in AIDS patients because the parasite just recrudesced in the brain and would cause encephalitis, and it was a very difficult control. There's no good drugs to cure you of toxo, and there's very few drugs that they can use to try and control parasite multiplication. So any patients that are immunocompromised, you see in cancer patients as well, toxoplasma can be a big problem. And I've got a picture here of a lesion in the retina of the eye. This is quite common with toxoplasma. You see ocular disease. And the interesting work coming out of South America, which made people sit up quite a bit, was that immunocompetent people were presenting with blindness and issues with toxoplasma infection that we really didn't see in Europe and North America. And this is probably, we think, due to some atypical strains of toxo that are coming out of South America they're much more pathogenic than some of the strains we have in the UK. So this, as a result of what was coming out of Brazil and some of the research coming out of Brazil, there was a big report by the Food Standards Agency a few years ago where they suddenly got a bit more nervous about something they really hadn't paid too much attention to because they didn't really understand what strains of toxoplasma were out there, what the risk was to people from picking up toxoplasma from food. That's from undercooked meat from these intermediate hosts. And they put forward that toxo really should be a priority for control because there was other studies coming out of the Netherlands looking at how outdoor reared animals, especially pigs, are much more likely to be infected than pigs kept indoors. So it suddenly became a bit of much of a higher risk. Also, some of the work around the behavioural studies that I talked to you about before really hit the headlines. And this parasite got the front 5 pages of the Independent, again, a year or so back, on the back of that Food Standards Agency report, which was really brought it to the fore. It's also quite good for grant applications if your parasite is on the front 5 pages of the Independent. So, but it really brought toxoplasma to the fore. Now, the other thing was they wondered, there's a vaccine that we use in the veterinary field that we helped to develop at Moredun quite a few years ago now. And people didn't know whether that vaccine would actually be helpful in terms of trying to prevent that transmission in food. And there was also no accurate prevalence data for the UK. We didn't know how many people were infected. About the transmission, that cats are really important in this. So members of the cat family, the sexual cycle of the parasite takes place in the gut of the cat. Just a picture of it here. When the cats usually go out hunting for the first time, young kittens, they'll pick up infection from birds or mice that are infected with toxo. And in the gut, you get the development of the parasite. The cats will shed the oocyst, that's the egg stage, in the feces, and they'll shed millions and millions and millions of these at a time. They've got a very tough outer shell. all warm-blooded animals. We can pick up the infection either from consuming these oocysts, from contaminated food or water. Once the parasite gets inside you, it starts multiplying inside cells, and these are the fast-replicating tachyzoite stage that's inside the cell. And using this, the parasite will disseminate all through the body. And on the pressure from the immune system, the parasite will differentiate into a tissue cyst. That's it here. And you can see the cyst wall on the outside, and inside you can see They like to go in the brain, so they tend to sit in the brain for the lifetime. They also go into tissues. So if you eat undercooked meat containing T.gondii cysts, you can pick up the infection that way as well. And the other way, as mentioned before, is this congenital transmissions, which is a vertical transmission from the mother to the fetus, only during a primary infection. So foodborne toxo. If we look across the world, looking at different statistics, most people do seem to acquire the infection from eating undercooked meat that's infected with the parasite. And there's some new diagnostics where you can actually look at the serology of people after acute infection and see if they've picked it up from oocysts or from bradyzooids. So it's quite a useful way of looking at where is the major issue. I mentioned to you before that outdoor reared food animals that are quite fashionable at the moment for welfare reasons are much more likely to be infected with toxo. And if you take into account the impact of the disease, and that's largely based on the congenital infection, and although the incidence in women is not high, the impact of having a congenitally infected child is massive, and it's lifelong. And if you look at disability-adjusted life years, which is one way of looking at impact, toxo does come out as one of the most important foodborne pathogens, higher than some of the others that are of higher incidence. So because of this, people are taking a lot more notice of it. We did a survey a year or so back of sheep across Scotland. We looked at 125 flocks, and we looked at what the prevalence was in the flocks. And you can see they're from all over Scotland. And every single flock we looked at had at least one positive sheep. And the overall prevalence was 52%, which is high, which I think shows you sheep can only pick up the infection from eating oocysts from the cat. So it shows you the widespread contamination in the environment of oocysts. There was a recent study done in pigs on the back of that Food Standards Agency report, and they didn't separate indoor and outdoor reared pigs, but it showed that about 7.4% of pigs were also positive for toxo. So we're involved again in a European-wide study because they're very interested in looking at the prevalence in food animals across Europe. What we're trying to do is use very similar technologies and methodologies so we can compare with different countries. Looking at these different food species, even horses are a food species in Europe, accidentally here. We're also looking at harmonising methodology to make sure that we can compare results across Europe. And one of the things we're interested in is what are the looking at the anatomical distribution of T.gondii in the different tissues, particularly obviously the edible tissues, just to see what the risks are. That study's ongoing at the moment, and we're focusing particularly on cattle, which again was a species that we thought before on the veterinary side was not an issue for Toxoplasma, but it is a lot more now because we realise that it's a problem in public health, although maybe not in the veterinary side. So wildlife and the environment. We were very keen, again, to see what's out there in the environment in terms of these genotypes, just to see, have we got anything similar to maybe what's happening in Brazil, or are the strains we have in the UK different? And again, our student Alison went out and she did quite a big survey looking at wild carnivores across UK. And we got a prevalence there of about between 6 and 40%, depending on the species. And she's been doing some genotyping on those samples. So far, the genotypes she's found have been very much related to ones we would normally find in Europe. We haven't found many atypical genotypes that you would find over in South America. The other thing that, again, we found quite interesting and looked at was the oocyst, again, widespread oocyst contamination of the environment coming from the cats. I'm going to finish up. I'm giving you quite a big sort of background there to what the issue is, to look at what prevention and control strategies we can do. We have been doing a lot of work on the veterinary side, looking at vaccination, really to prevent congenital infection in sheep and goats. And what we were very interested to look at, again, working with some of the public health groups, so could we also vaccinate food animals to prevent the development of these cysts and therefore try and protect public health? The one that I think would be a really good one to do would be to try and prevent oocyst shedding in cats. That would be the key one. So to summarise what we did there, outdoor reared animals are much more of a problem for infection. Vaccination definitely did reduce the tissue cyst burden. And there were differences in the risk of infection from the different tissues. So I just want to finish up by thanking our group here. I was really the spokesperson today for a lot of the work that's been done in our team in Edinburgh. And I particularly like to acknowledge Frank Katza, Alison Burrows, our PhD student who did a lot of the work on the wildlife and the pig vaccine work, and Jean-Luis Garcia, who did a sabbatical year with us from the University of Londrina in Brazil. So thank you very much for your time.